friends, today we are going to discuss about the second chapter of 12th science, biology, and that is reproduction in flowering plants. We know that reproduction is the process by which the young individuals, after attaining maturity, they give rise to new individuals. Reproductions are of several types, but the main favorable one is known as sexual reproduction. Sexual reproduction takes place by fusion of gametes. Gametes means reproductive cells, and those gametes are produced by a single organism or by two different organisms. Now, we know that the flower is the reproductive unit or reproductive organ of plant. That is flowering plant. Flower is having two kinds of roles. The first one is known as calyx and another one is known as corolla. They are known as accessory floral roles because they do not take part in sexual reproduction, but they help to attract the insects for pollination. Androecium and gynoecium, they are known as reproductive floral roles. Androecium is known as male reproductive floral role. Gynoecium is called female reproductive floral role. Now, androecium is made up of stamens and gynoecium is made up of carpels. So today we are going to discuss about the structure of stamen, the structure of anther, the structure of pollen grain, and the development of male gametophyte before release of pollen. So now, friends, if we see the structure of stamen, here in the diagram you can see the stamen is made up of two main parts. The first part is a slender stalk-like region which is known as filament, and the upper sac-like structure which is known as an anther. The joining of filament and anther is known as connective. The proximal part of filament is either attached to the petal or it is attached to the thalamus of the flower. Now when we see the structure of the anther itself is a sac-like structure. In most of the flowering plants, that is angiosperms, the anther is having two lobes, that is bilobed, and every lobe has two theca. So we can say the mature anther is bilobed in nature. Every lobe contains two theca. That means it is tetrathecous in nature. If we see the transverse section of the anther is having four corners. That means every corner contains a part that is known as theca. So, an anther is a tetragonal structure. In every corner there occurs a theca, and the theca contains Microsporangium. And this Microsporangium, which is there in the theca, it is longitudinally arranged to the length of the anther. Now, this microsporangium will again develop and it will produce number of pollens. So when we see the internal structure of microsporangium, the outermost surface of microsporangium is circular and it is made up of 4 different kinds of layers along with some tissues inside it. The outermost layer is known as an epidermis. The second layer is known as endothecium. The third layers, they are known as middle layers, and the innermost layer is known as tapetum. The outer 3 layers, epidermis, endothecium, and middle layers, they are protective in nature. They protect the inner tissues of microsporangium, as well as they are responsible for the dehiscence of anther. Dehiscence, that means breaking of anther to release the pollen grains. Innermost layer, that is known as tapetum, contains the cells which are responsible to provide nutrition to the developing pollens or microspores. Now, on the inner side of the tapetum, sometimes very compactly arranged mass of similar cells are present, and those cells constitute a tissue, and that is known as sporogenous tissue. The cells of sporogenous tissue, they are diploid in nature, and these sporogenous tissue divide mitotically and then meiotically to form pollen grains or pollen tetrads. Now what will happen? The sporogenous tissue undergo division, mitosis, and they will produce number of cells which are known as pollen mother cells or PMC. The pollen mother cell, they are again diploid in nature. The pollen mother cell undergo meiosis, and after meiosis, every pollen mother cell will give rise to 4 microspores, and they are known as pollens. But initially, when they are not mature, all 4 pollen grains or microspores remain attached to each other, and that is known as pollen tetrad. But after attaining maturity, after dehiscence of anther, they become dried up and they are liberated, they become free, they become round, and now they are known as pollen grains. Now friends, we are going to see the structure of typical microspore or pollen grain. As we have seen in the structure, or you are having the NCERT textbook, in that NCERT textbook also you can see the pollen grain is a microspore. It is having 25 to 50 micrometer of diameter. The shapes of the pollen grain may be round, may be oval, may be oblong, may be roll shaped. The outer surface of the pollen grain may be either smooth or spiny, depending upon the type of pollination. The pollen grain contains two layers, an outer layer and the inner layer. The outer layer of pollen grain is known as exine. The inner layer is known as an enzyme. The exine is made up of specialized structure that is known as sporopollenin. And this sporopollenin is the structure, is the material, is the chemical which cannot be degraded or decomposed with the help of either strong alkali, strong acid or high temperature. But the scientists do know that not a single enzyme is present which can decompose the sporopollenin, and because of the same reason the pollen grain are observed, are seen, are obtained in the form of fossils. Now the inner side, the inner layer is known as an enzyme. An enzyme contains either cellulose and or pectin. Now if you see the outermost surface at some of the places on the surface of pollen grain, sporopollenin is absent. And those places are known as germ pores. Germ pore is the region from where the pollen tube will develop during the germination of pollen grain. That means, because after pollination, when the pollen grain will reach to the stigma of carpel, the pollen grain will germinate, a pollen tube will develop, and that pollen tube will come out from the part which is known as germ pore. On the inner side, when we can see a distinct nucleus and plenty of cytoplasm. During the development of pollen grain, what will happen? The nucleus of the pollen grain will undergo unequal division, and so that two nuclei are produced. One will be larger, another one will be smaller. One will be larger, another one will be smaller. The cell with larger nucleus is known as vegetative cell, or it is called tube cell, and the smaller one is known as generative cell. The vegetative cell is having abundant food material. It is having irregular shaped nucleus, and the smaller one, that is generative cell, is having spindle shaped structure, and it floats in the cytoplasm of the vegetative cell. Now, friends, let me discuss some more things about pollen grain. The pollen grains are somewhat allergic in nature. If you have studied the Congress grass, if you have heard about Parthenium, the Parthenium or the Congress grass or the carrot grass, when the pollen grains are released from that, it can cause asthma, it can cause bronchitis. Some of the allergic reactions are observed in the body of human beings. But in some of the cases, the pollen grains are also used as a food material to provide energy to the race horses, to the athletes, etc. Syrups are being made, the tablets are produced from the pollen grains. So pollen grains are almost important to our healthy life. Now, when we talk about the viability of pollen grain, That means when the pollen grains are produced, the pollen grain, by the process of pollination, they will reach to the stigma. So, before entering the reproductive phase, that means when the pollen grains reach to the stigma, they must be viable, they must be living. And what is the viability period of a pollen grain? It varies from species to species. It depends upon the moisture, it depends upon the biological factors or environmental factors. As for example, in wheat, rice, etc., when the pollen grains are released from the mature anther, the viability is only 30 minutes. Whereas in the families like Solanaceae, Rosaceae, Leguminosae, the viability of pollen grain ranges from days to months also. You have studied or you have heard about artificial breeding. In same way, the pollen grains are also stored, they are preserved in minus 196 degrees centigrade in liquid nitrogen. The process is known as cryopreservation, and these pollens are used to establish the pollen bank. And these pollen banks are also responsible for the establishment of gene bank or seed bank for the cultivation of crop plants. So that is all for today. Today we have discussed about general aspects of sexual reproduction in flowering plants. We have discussed about the structure of stamen, the structure of anther, the structure of Microsporangia, the process of formation of pollen, that is microsporogenesis, the structure of pollen, and the utilization of pollen. In the next lecture, we will discuss about the carpel. We will discuss about